Hi everyone, welcome to the third Bats in Churches live session. Thank you very much for joining us. So my name's Claire, I am the Training and Survey Officer for the project and I'll be the host for this session. So today we are returning to the wonderful world of bats, and over the next 45 minutes we'll be learning a bit more about the discoveries about bats and the way they live through the ability to be able to research and study their genetic material, their DNA, the building blocks of life. And to do this, we're joined by two fantastic speakers who'll be able to share the knowledge of their research and their batty experiences. So we're very happy to be joined by Professor Fiona Mathews from the University of Sussex. She has a wealth of knowledge about bats and is the chair of the Mammal Society and also the co-founder of Ecotype Genetics. And also the fantastic Megan Power, who is a third-year PhD student with University College Dublin and also the University of Bristol. And she'll be talking to us about her really interesting research into greater horseshoe bats in a population in Gloucestershire. There will, of course, be time to ask questions. So please do get your questions coming in for Fiona and Megan using the Q&A function on Zoom, or through the hashtag on Twitter, Bats in Churches Live, or via Facebook if you'd prefer. Just so you know, Diana Spencer, one of the engagement officers for the project, is in the wings, ready to answer any kind of tech issue questions coming in, or any questions related to bats in churches. So Bats in Churches has actually made some use of DNA analysis of these molecular techniques. So part of the project is a research study, the Bats in Churches study, and we're asking volunteers to take part. So you can find out more about that on our website. But we're actually taking bat droppings from different churches and sending them off for DNA analysis to find out which species of bat are using the church. And that's one of the tools that we're using to help understand how bats are using churches and whether this changes for different species. But of course, these molecular techniques have a wider role, and we can find out a lot about bats this way. So without further ado then, I'm actually going to hand over to Fiona, who will be able to share more about this and her experiences and research. So over to you, Fiona.
Thank you. I'm just going to check that you can see my screen. Yes, brilliant. Well, hello everybody. Bats obviously are one of the most challenging of all our mammals to monitor. For most people, all we manage to see is sort of a glimpse out of the corner of our eye, and it's very difficult to identify what species it is, let alone how many of them there are. And what I'm going to do in this talk is give a bit of an overview about the importance of molecular methods to studying bats generally and understanding their conservation. And then I'll tell you a little bit more about the work we do at Ecotype Genetics to finish off. So just over, well, actually it's 2 years ago now, I led a piece of work that reviewed the population and conservation status of all Britain's mammals, and this was the first time this exercise had been done since 1995. And one of the things that immediately struck me in doing that work was, despite me setting out thinking I know a bit about bats, that's going to be my easy bit to write, actually it turned out to be a complete nightmare, and it was by far the hardest section of anything. Since then, this is hot off the press, we've also at the Mammal Society have just brought out a new atlas of Britain's mammals, and in that are pictures like the one you can see on the right, which basically identifies for each grid square in the UK whether or not a species is present. And at the moment, those sorts of maps only make very small use of genetic data, because essentially most records that go into biological record centres are still based on observations that people make or acoustic records. So I'll tell you a bit more about some of the problems with that as we go. If we think just a little bit more about bats, basically out of Britain's 56 species, 32% of them are bats. And in terms of what that means for the mammal fauna, just thinking about England, we've got roughly 155 million animals, we reckon, give or take a bit, of which 148 million are native, and about 7 million of them are bats, which is about 5%. But I'd point out to you that there's a huge caveat around that number of 7 million. And one of the reasons is that it's just very difficult to get good data on where exactly bats are, and secondly, how many of them there are. If we look at how they've been doing recently and try to, we've done this for all species, try to look at how their range has changed over time, greater and lesser horseshoes are doing well, grey long-eared are going down, Daubenton's seem to be about the same, but look, all these other bats are ending up in the we really don't know what's happening category. And part of the reason for that is while we have some increase in numbers of dots on maps, we also know that there are quite a lot of issues with that to do with them being based on detector records with different degrees of verification and so on. In terms of population, greater and lesser horseshoes seem to be going up. We're not certain that anything is going down or staying the same. And if we look at the don't know, well, we've got again this massive list of, in terms of population trends, we really have very little understanding of how things are changing over time. And for the species in brown there, we weren't able to make population estimates at all. So that's not even the first stab at how many of these animals we have. So if we're trying to understand the importance of populations living in churches, you can see it's really difficult to do that without any wider understanding of the population size as a whole. So the final part of the exercise of reviewing Britain's mammals was to come up with a red list. And this uses exactly the same set of criteria that would be applied to rhinos or tigers. And essentially it ranks animals according to the degree of imminent threat. And if we look at what's going on with bats, we've got grey long-eared coming out as endangered, serotine, which often uses churches, coming out as vulnerable. And I'm just going to skip to this section on the right, the data deficient. We've got these small myotis, whiskered brants and alcathoe, where we weren't able to say anything at all. And in red list terms, that's seriously bad news, because everything about the red list is set up to make you put it into a category, because the data deficient bunch tend to get kind of swept under the carpet because we just say they're too difficult, we don't know what's going on. But we didn't even know what their range was, let alone anything else. So we've got some really major research questions here that I think we need to get to grips with. And again, just coming back to the churches, we know that at least whiskered and brant bats can and do use churches. And so it could be a really important place to start looking at what species we have and their distribution across the country. Okay, so why are we data deficient, despite obviously a lot of effort goes into trying to understand the population and conservation status of bats? One reason is that a lot of surveys are heavily dependent on acoustic methods, and for those species that end up in this data deficient category, a lot of them are difficult to distinguish based on acoustic data alone. So myotis bats, for example, is very difficult. Or if you want to separate out a grey and a brown long-eared bat, you're going to be really hard pushed to do that using acoustic means. And if you're looking at, I don't know who contract ecology are, I should say that, I just picked this off the internet. But you know, if you're looking at a complex structure like a church, it becomes extra difficult to understand how many bats are there, just because there's so many potential exit points, and it's really a very challenging thing to do. Even just to establish presence absence is tough. And then on top of that, we've got the really annoying thing that quite a lot of these bats, even if you manage to get close and personal to them, really hard to distinguish, even for experts who've got them in the hand. So at the top there, we've got a grey and a brown long-eared bat, and on the bottom we have an alcathoe and a whiskered. And you know, I can't emphasize enough how tricky these are, and really you are relying on those clues from molecular analysis to help you out in distinguishing what animals you have. And we have actually analysed one of the bats in churches, samples from Devon, and found a grey long-eared bat in a church. Yay! Which is really very, very exciting. Now, bats do leave clues to their presence. Actually, manage to physically spot them. They do leave droppings behind, and I'm not in any way underplaying the difficulty sometimes of finding droppings when they're in awkward to access places, but nevertheless we know we can and do find droppings in churches. And historically people spent a lot of time looking at them, including using a microscope, but looking at their morphological, morphometric characteristics to try and work out what species they were. I'd say that there's a lot of art to that, and a lot of overlap. So for example, small bats, juvenile bats have smaller droppings than adults, for example. So if you're in a maternity site, you might be misled by the fact you've got multiple sizes of droppings in. Also, there's a lot of overlap in, there's rather few species that you can say definitively, this is definitely a species just on the shape of its droppings. Okay. But happily, droppings do leave us some telltale clues, which then we can use for our molecular analysis. Okay, so I've been working on issues around molecular biology of bats for about 10 years or so, and started off mainly doing population genetics, so to understand things like how inbred populations are. We then got asked by Swift Ecology if we'd like to take over the work that they've been doing with Waterford, who had been providing a service, quite a simple service in genetic terms, really, just identifying what species are present on the basis of droppings. So I teamed up with my colleagues at Sussex, Tony Carr, who's Professor of Molecular Genetics, and Dr. Tom Etheridge, who frankly does most of the work. And we set up this little lab, we set up a company, and where we do basically DNA-based ID of bats and other things as well, but primarily bats. And what we're doing there is basically people send us samples. Our job then is to get the DNA out of that, which is actually fairly straightforward with the process of a buffer. We then amplify the DNA that's there. Just before I go on, I'd say one of the really important things is to avoid contamination in the lab, because we want to be sure that when we send back a result, it really is what we think it is. So we only ever deal with one sample at a time, and we disinfect the work surfaces in between, and everything's done in a sterile hood. And then what we do is we do real-time PCR on it, where basically we're splitting apart the DNA and then put in primers that are labelled with fluorescent dye, so that when the amplification steps are going along, what we're getting is increasing amounts of this dye in the product. And we make these primers are specific to particular species. So because we've got this alphabet soup, if you like, in our little test tube, we can put in specific primers that have exact matches that will only match one particular bat species. And then we can look at these fluorescent products being made. And what we end up with, I'm not going to go into the detail, but basically this is where we've tested for a single species, and we get this curve coming off as we get more and more of the fluorescent dye in our product. Okay, and in these replicates we put in the machine, you can see that 33 and 34 didn't have anything at all, and then we've got this hit at 35, and 36 had nothing. Okay. One reason why we use real-time PCR is because cleverly it lets us look at multiple species at the same time. And this is something that personally, coming from a background of doing a lot of bat work, I think is really important, because I think very often we do have an assemblage of different species with the roost, and while a cursory look may lead you to think it's only one species, you might think you've only got long-eared. Actually, very often you've got other things in there as well. You've often got the small myotis mixed in at the same time. So what we can do is we can put in these different primers that are specific. They've got specific labels for, so for example, we look at ferritin, whiskered and natterers all together. we can test for multiple species at the same time, and then we can get multiple curves coming off for different species simultaneously. So for me, that's a big advantage of doing this PCR-based approach, and it also means that our assays are very specific, because we know that we've got the exact, it's a bit like having an exact key that only fits one lock. We know that when we're amplifying something, we've got what we think we have. And if nothing works, it's very clear. So this is the case, nothing has worked. So you can see nothing is amplified in there. If nothing works, there's a couple of reasons why. It could be that the DNA is just degraded, and that is an issue with bat samples, particularly damp. It could just be that there's very, very little DNA in there for us to work with. It could be that what has been sent to us actually isn't a bat. And one of the things that we do at this stage is we then go to sequencing. So we could send away, we sequence a particular part of the genome, and that will tell us, for example, if we've got a shrew. We get remarkable numbers of shrews, actually. And it's amazing how many bat workers do send us what turn out to be shrew samples. The reason why, in case people are wondering, the reason why we don't go to sequencing straight away is the problem with it is it will give you the first hit that it comes up with. So basically it compares your sequence with a genetic database, which in most cases is fine, but if you've got a mixed sample, it basically is going to come up telling you what the primary thing in your sample is going to be. So it misses out this extra step. Okay, so to summarise, important homes to some of our conservation priority species. I'd highlight particularly grey long-eared and serotine bats amongst there, and also the small myotis, the whiskered brants and alcathoe, that we really know very little about. Using DNA, we can establish what species we have without physically having to see them or have them in the hand. It's really the only reliable way to distinguish some of these cryptic species. And also, by doing the DNA analysis, we're getting vital data that can really help contribute to our understanding of conservation trends for bats nationally. Thank you.
Fantastic. Thank you so much, Fiona. And that's really cool. If so anyone taking part in the Bats and Churches study, you now know what happens to your collection of bat droppings when you send them off. So that's really cool. Fiona, actually, there's a couple of questions here specifically related to your talk that I do want to just ask you quickly before handing over to Megan. So there's one from Les Bentley who asks about the reliability of these tests. I just wondered if you wanted to.
Yeah, I would say that they're extremely reliable. Obviously, we get the cases where it's just not possible to get an answer for you because of degradation and those sorts of things. We've done a lot of work looking at whether there can be cross-binding. So can there be cross-reactivity? If you imagine like the key in the lock analogy, how likely is it that the key might accidentally unlock a different door? And we've done a lot of work on that and looking at cross-reactivity. And initially when we started out, we had an initial problem that was flagged with a serotine bat giving a weird result. And what we did is we just completely redesigned the primers. So we haven't had any issues of cross-reactivity that worry us. Once you're at sequencing level, again, most of the time it should be correct, but I don't know if Megan wants to come in, but you know, occasionally people can get really curious results, most often due to contamination. So the key thing is if you're doing this, make sure you're working with a lab that has really clear protocols for avoiding contamination, because that's the main. It's not so much that the assay is going to be wrong, it's more likely that you've got contamination somewhere along the pipeline. So the sort of things we do is we have filters in all our pipette tips so that we can't accidentally contaminate from one sample to another. We do everything in a protective hood. We actually do our amplification step in a different building from where we do our DNA extraction, because you don't really want to be working in a lab where you've got lots of DNA from bats kind of in the atmosphere. So we avoid that that way. And the other critical thing is also to think about the collection protocols, because very often, you know, bat workers will have had maybe pots that are swooshing about in the bottom of their rucksack that they then fish out to put a sample in, or they might forget the fact that actually you don't want to have gloves on that you've been crawling around another part of the roost in if you're trying to give us something that's meant to be specific for one area in your church, you know. So I think those sorts of contaminations could also be a problem.
Are there any top tips? We have a couple of questions about how to preserve droppings for DNA analysis, and also whether the age of the droppings will have an impact.
Yes. Okay. So first top tip is to put your samples in some sort of rigid container. And we're always really happy to send out little tubes that you can use to put them in, because the worst case scenario for us is where we get a pulverized mess in the bottom of the tube, just because it makes it a lot more difficult for us to extract all the sample from there, and also to see what's going on. In terms of preservative, when we first set out doing this 10 or 15 years ago, we used to tell everybody to put samples in alcohol. We no longer recommend that people do that, because actually what it does is in the feces there are inhibitors as well. So there's inhibitors that will stop the PCR reaction. And it seems that actually you make that worse by putting the alcohol in, and the best thing to do is just to air dry it, or you can put some silica beads in it if you wish, but basically normal drying is fine. What you really don't want is for your samples to start going mouldy, so you do need to make sure that they're dry. In terms of how fresh they should be, the worst or the most difficult samples for us to process are actually ones that are fresh out of the bottom of a bat. And again, it seems that there's some issue there with inhibitors, and which is why again we've gone to the air drying protocol, which seems to stop that problem. Over time, so if we're talking over years, DNA will certainly degrade. But the whole way we set up the process is that we have a long series of amplification steps. So with enough effort, normally it's possible for us to get DNA out of stuff if there's anything there to be got. You know, we can't guarantee 100% of the time, but you know, and you think you can manage to get DNA out of a meal, you know, fossilized stuff. Yeah. So
perfect. Thank you so much, Fiona. We've got a few more questions. We've got quite a few questions coming in actually, but I'm actually going to hand over to Megan now, but there will be a time to ask Fiona some more questions at the end. There's one actually here about other materials that you can use. Is there anything else apart from droppings? But I think Megan will be covering this a little bit, because she doesn't actually
Can I say something quickly?
Oh, please do, Fiona, please do.
Which is, I've been doing some work looking at using wing swabs. Partly this is more with a view to doing population genetics work. So the problem with fecal samples is the DNA does get fragmented in those, so you get very short runs, which means it's not so suitable for doing population genetics work. We can use it fine to identify species. And so I started just playing about myself using swabs as opposed to taking wing punches, which obviously the wing punches require a Home Office license. And that's been looking really promising. And actually the picture of the Alcathoe that I sent you, that I showed you on the slide, that Alcathoe wouldn't actually do a poo on the night. They had it bagged up for ages, didn't get anything. And so that was a sample that was sent to us using, it was actually a homemade swab, but it worked, and we managed to amplify stuff from it.
Thank you. I can't believe it didn't perform for you on the night, Fiona.
Wasn't easy, was it?
Okay, I think I'm just going to pass over to Megan, who will be talking to us about her work with greater horseshoe bats, looking at the effects of different life strategies on telomeres, and she will be able to tell you what that means. Brilliant. Thank you, Megan. Over to you.
So is the screen sharing okay?
Yes,
yeah,
it
perfect.
is. Thank you.
So I'm just going to talk to you a bit about my PhD research and then some of the things we can learn about bats through their DNA. So I know it was covered in kind of the first webinar, but we know that bats represent 20% of all mammalian species, and recent taxonomic revisions have found that there's now over 1,400 species. So there's actually been a 25% increase since 2005. So we know they're the only mammal capable of true powered flight, and they're found on every continent except the poles, and some use echolocation as a sensory mode to find prey, navigate in darkness, and mate. However, they do also have some very unique life history traits. So one of the most unique is that they're some of the longest lived mammals given their body size and can live up to 10 times longer than expected. So when we consider longevity quotient, which is essentially correcting for body size, we find that 19 species of mammals live longer than humans. So one of these is the naked mole rat, but then the other 18 species are actually bats. So they occupy several dietary niches, so many being insectivores and frugivores, but we do also have some unique species that are carnivorous, as seen here with the fringe lip bat, and we all know the kind of the Halloween, some feed on blood, so the vampire bat. So while smaller bats typically have one offspring a year, you do have in different parts of the world some that can have twins and triplets. So example, eastern red bats. And with bigger bats, have like flying foxes can have 2 reproductive events a year. However, as mentioned with the previous slide, bats can live an exceptionally long time given their small size and high metabolic rate. So we now know that the oldest bat ever recorded was a 7-gram Myotis brantii, which was recaptured at 41 years old. And what's so interesting about bats is that they really show little signs of aging, and there's very few age-associated diseases rarely reported in bats. So aging to us is at least the most interesting topic in biology, but it's also one of the least understood. So I think to further understand more about mammalian aging, I think we need to understand the specialists, which in this case is the bats. So there have been 9 hallmarks of aging identified. However, these have mostly been obtained from short-lived model organisms. And so it's quite hard to really determine if experimental findings from short-lived inbred model organisms can be translated into long-lived outbred mammals such as ourselves. And I'm going to focus in a bit more on one of the hallmarks, and which is one of the best characterized, which is telomere shortening. And as Claire said, probably like I'll introduce the topic of telomeres. So what exactly is telomeres? So what telomeres are is they're repetitive sequences of DNA that essentially cap the ends of our chromosomes. So I always kind of like to use the analogy, you can kind of think of them as the caps at the end of your shoelaces that prevent the shoelace from unraveling. So as our cells divide, the machinery can't reach the end of the chromosome and can't replicate to the end of the chromosome. So telomeres get shorter each time a cell divides. So once they become critically shortened, the cell stops dividing and undergoes what we call senescence. So what this means is the cell is still metabolically active, but it can no longer replicate. And aging is thought to be caused by an accumulation of these senescent cells as we get older. And so what about bat telomeres? So telomeres have been well characterized in other species. There's still not much known about telomeres in bats and with age. So in a paper published in 2018, cross-sectional data actually showed across 4 long-lived species very different patterns. So shortening was found to occur in 2 long-lived species, so the greater horseshoe bat and the bent-wing bat, while no shortening was reported in 2 species of Myotis, so Myotis myotis and Myotis becksteini. So this gives us an indication that even in the longest lived bats, they probably utilize different mechanisms to maintain their longevity. And this could potentially be down to different life histories. And this makes sense as recent analysis has shown that extreme longevity in bats has evolved across the family tree at least 4 times. So it's important to gain an in-depth understanding into these different patterns in relation to telomeres and aging in bats. And this is where some of my research comes in with the greater horseshoe bat, Rhinolophus ferrumequinum, a bat that has some very interesting life history traits. So they have a maximum lifespan of 30 years given their small size. So they're normally about 22 to 26 grams. So they have a polygynous mating structure. So in this population, we do know that some of the females visit the same males over a number of years. And so a bit of background on the population I work on, it's been extensively monitored by Dr. Roger Ransom since 1959 in Gloucester in the UK. So even when he was a school teacher, he used to cycle down to Woodchester Mansion on his lunch breaks in order to collect data. So this makes this population really unique and really interesting to explore trade-offs in aging and looking at telomeres. And just a reminder that reproduction is only one part of a really complex cycle. So horseshoes can undergo deep and prolonged torpor in the winter, and this has been shown to be positively correlated with maximum lifespan across bats that do hibernate. So how exactly do we look at telomeres? So we simply take 3mm wing biopsies from the individuals. So as Fiona said, it does require a home office license, but it's quick and simple, and at least we know the wing biopsy site does heal quite fast, and there's been no adverse effects on any of the individuals. So sampling has been ongoing since 2016, and at this stage, from nearly 600 individuals, we've nearly 2,000 samples, and so this allows us that we have telomere sampling from different points of a life of an individual. So samples are primarily taken from 2 sites. So the maternity colony, which is located in Woodchester Mansion, and then also the hibernaculous sites, which are located in Nailsworth Mines. And so essentially this allows us to cover the really key periods such as early life, hibernation, and reproduction. So kind of what have I found so far? So as with the previous work done in cross-sectional carried out in horseshoe bats, we find even with tracking the same individuals year after year, there's still a decrease with age, with the biggest decreases appearing to occur really early in life. However, when we went down to the individual, so within individuals themselves, we found that shortening came up as non-significant, and we were kind of a bit baffled by this until we then kind of just simply had a look at telomere over time and found this. So in contrast to many organisms that show a consistent pattern of shortening with age, humans being one of them, we found that horseshoes didn't have such a simple pattern. So while the telomeres ultimately shortened with age, we found that they're showing these sporadic elongations over their lifespan. So while they ultimately shorten, we potentially think that as perhaps a link to their longevity is being able to modulate their telomeres. However, to fully understand if telomeres are able to elongate and during what part of the life cycle is particularly important, and is this related to any potential trade-offs? So for this, we need to dive really into critical life history stages. So here's an example of some work carried out in early life. So given the population, we're able to catch babies from when they're born up until their first year. And what we found was the really early life decreases are really rapid in the first 40 days of a pup's life. And this is likely due to the rapid growth observed in the species and given the high number of cell divisions required for the body to grow. So in this, so we found that pups reach approximately 90% of adult size by 45 days. But yet from 40 days onwards, we start to see increases in telomere length from 40 days onwards, and also great variability between telomeres of individuals of the same age. So in a unique case where we're able to take cohorts, this is a cohort that were born in 2017, and we were able to track them through the year through telomeres, we found that over hibernation, this is primarily where elongation of the telomeres occurred back to birth levels of telomeres. Telomere length. So it appears that pups are capable of recovering from the effects of growth and potentially also the environment. So it could be that investment into these maintenance systems like telomeres could increase potential of adult survival and future reproductive events. And so while telomeres do ultimately shorten in the horseshoes, perhaps modulating their telomere length between stressful events could potentially contribute to their unique longevity. And we find that hibernation is really key because even while it's not shown here, adults seem to show a stable pattern with no massive change over hibernation. So hibernation seems to play such a massive factor. And another key aspect of the life cycle is reproduction. So this is a unique population in terms of, even though bats can start breeding from 2 years old, not all of them choose to do so or given environment, and they can delay to when they're 3 or 4. And what we found was that females who started breeding earlier had a greater telomere shortening rate than those that actually delay their breeding until when they're 3 or 4. So this is interesting because it ties into work that Roger carried out in the early 90s, where he found that females that bred earlier had lower survival rates than those that started breeding later. So this is interesting because it kind of shows nearly individual strategies and potentially this reproduction lifespan trade-off, which is quite good. It was also shown that females who bred consistently each year also had a greater shortening rate. So it kind of shows that the pressure of reproduction, given that pregnancy and lactation are so energetically expensive to bats. However, as well as kind of my project, I have also been involved in some other things. And one of these is there's many other ways in which we can understand bats. And one of these is the field of genomics. So why study genomics and what can it teach us? So a genome is an organism's complete set of DNA. So it basically contains all the information needed to build and maintain an organism. So you can almost think of it like a blueprint. So sequencing genomes are quite an important step to, one, understanding genomes, and then as well with sequencing animal genomes, allow us to reveal the genetic basis between specialization and adaptation and to understand molecular mechanisms behind such differences. So the first human genome was fully completed in 2003, and so this comprised of 3 billion DNA bases, and at the time was estimated to a total cost, I think, 2.7 billion. However, as we're living in a technological age, the cost of sequencing has reduced. So it's now possible to sequence a genome and analysis for $1,000, with predictions that within the next 5 years, this could potentially go down to $300 per genome. And I suppose, why is this important and how does it relate to bats? So recently, one of my supervisors, Professor Emma Teeling, has launched a genome consortium known as Bat1K. And this key goal is to sequence the genome of every living bat species, which is no small feat given that we now know there's over 1,400 species. And this is to further understand aging, echolocation, and many other adaptations within bats. So what exactly have we learned so far? So we recently undertook a pilot study where we were able to sequence 6 bat genomes across different families. And this yielded many interesting results. We're currently just still in review, but what we found was, in contrast to other mammals, there was various gene gains and gene losses in genes related to immunity. So this indicates that bats have evolved, these molecular mechanisms to allow them higher tolerance to pathogens. There's also evidence that, the origin of echolocation, that echolocation evolved once in bats and was subsequently lost in some species, and that there was also unique viral integrations found in the genome. And so what these integrations are indicative of is past viral infections, and allows us to highlight, viruses that bats have co-evolved with and tolerated, and gain a better understanding. And so to currently advance on this work, we're currently sequencing a representative from each of the 21 families. And this is to hopefully to gain further insight and to try and really tease apart the molecular mechanisms behind echolocation, flight and aging, and all the many other unique adaptations bats have. So I just want to thank the Bats and Churches for inviting me to talk at the webinar. I always have this kind of slide because there's so many people that wouldn't, this project wouldn't be able to go out ahead with. So my supervisors, Emma, Gareth and Roger, I'm really lucky to be part of 2 amazing labs in UCD and Bristol. And then a big kind of shout out to the volunteers that helped me and Roger in terms of collecting data for the horseshoe population, as it would not be possible to have maintained it this long without their help. So thank you very much.
Brilliant, thank you, Megan. Wow, nearly 2,000 wing punch samples.
Yeah, which
wow.
is, yeah. So it's been a big, massive effort, but what we found with the paper that had published previously on bat telomeres was from a member of my lab previously, and it was just cross-sectional, so it wasn't change within individuals. And what we found is that, with these longitudinal studies, is that it really gives us an opportunity to really examine aging and really examine life history differences. So if we're really well able to track individuals within this population, I know at the moment the oldest girl is 23, so it's really, unique, and you kind of see these bats and kind of see the changes they're undergoing.
Fantastic. We've got a question specifically about that, actually. One from Tricia Scott, who's asking from your presentation there about human telomeres and whether they show the same patterns in relation specifically to reproduction, greater loss of telomere length if they breed earlier.
So there has been a massive paper that was done recently, and it was actually using telomere length, and it was part of a Filipino cohort. So there was a lot of information where they found that you know, females that had more babies had started breeding, you know, started having babies breeding, started having babies younger in life, that they actually had a more rapid decrease in their telomeres. So humans, there hasn't been really any studies showing elongation. There is really the straightforward patterning. There is shortening with age, and that's it. But there's definitely different rates at which can happen. So it seems that reproduction really accelerates. And they found that even that the chronological versus biological age, that females that had more babies were actually biologically older than chronological counterparts. So unfortunately, having more babies makes you older.
Well, that's fascinating. Thank you, Megan. I'm going to bring Fiona back in as well, because we've got time for a few more questions, and we're getting quite a few coming in. This is actually probably best for you, Fiona. We've had a question from Philip Parker, who is asking about whether it's possible to sample several droppings at the same time. Basically, he's had issues with collecting droppings underneath a roost to confirm whether brown long-eared or grey long-eared, but it's actually come back as natterers, and there was kind of just one natterers using the church, for example. He's just wondering if there's possibility of basically giving you lots of droppings and kind of doing that at one time. Yeah. You see what I mean?
Yeah, so that's certainly possible. So you could send a pooled sample, and you know, so provided it's flagged to us that that's what's going on. Otherwise, what tends to happen is when you submit samples, this is not just to my lab, but generally people will tend to pick out just one or two and just take those and sequence them. So yes, it would certainly be possible if you think you've got a mixed sample and you think you've got brown or grey long-eared. Yeah.
Fantastic, thank you. Also, another one here. This could actually be for either of you. Do bats hybridize? Is there any evidence of bats hybridizing?
I'll let Megan answer that.
So it tends to be quite common sometimes between some of the Myotis species, so I know Myotis blighty, particularly, you know, along the continents, you know, of Eurasia. So yes, there is hybridization, and it can definitely make some of the species, such as cryptic species as we like to call them, in terms of when they all look alike, it can be quite difficult sometimes, you know, to distinguish between. So yes, hybridization does occur between some species.
Fantastic, thank you. And Ross Baker asks, do you know how many species have been fully sequenced worldwide so far?
Oh, it's hard because the problem with genomes is when the technological kind of, you know, earlier genomes would still be quite fragmented. So there has been a push lately to get more higher quality genomes. And so there is projects such as the VGP, which is the Vertebrate Genome Project, with the goal to sequence all vertebrate genomes. So there is very big undertakings. I do know of a study recently that had taken 130 mammal genomes, but in terms of an actual number, I wouldn't be able to pull a number there. But it seems to be becoming more prevalent now to at least try and sequence, you know, genomes of species of interest. But there is a lot out there and available.
Perfect. We've spoken a bit about droppings, about wing punches and wing swabs. We've been asked whether you can actually use fur and saliva. Fiona,
okay. Yeah, so saliva, it's not so much saliva as people have tried, as with humans, you can take cheek swabs. I wouldn't recommend it, mainly because there's a paper showing that actually you can end up damaging bats' teeth quite easily, because the rayon in swabs end up getting tangled around the teeth, and it's quite difficult to get enough cells on it. So I would not recommend that as a way forward. So species, different mammals are variable in how much DNA they have basically going up the fur shaft. So if you take something like pine marten, for example, the DNA actually goes quite a long way up the shaft, and you can get pretty good results from fur samples. Whereas for a lot of other species, you've actually got to pluck the fur out of the animal. So we have done this under Home Office license just to compare it. So you can get usable DNA that way. My personal experience of bats in the hand is they don't seem to enjoy having fur plucked. So I would stick with the tissue punch, the wing punch, which they don't seem to react to at all, actually, or a wing swab or droppings.
Perfect, thank you. And we've just got time for one more question. And just really that there'll be people out there, early career ecologists, that want to do the kind of research that you're doing. Could I actually just go to both of you just to just your advice about taking this on and this kind of career?
So yeah, just kind of follow, how I kind of got started with my PhD was that I actually did my undergrad project with Emma, and at the time, when I came back to her lab, she actually, there wasn't any kind of thing, there wasn't a job there specifically, but then I started working on the BAT1K, and that kind of led to kind of different opportunities and volunteering, and as I said, at least here in Ireland, doing the BAT walks and everything, and you'd be amazed by talking to the volunteers and just the people you get to know. So it's kind of, doing what you love, really, and just keep being involved, and if you see an event, just grab it, the opportunity to do it, and you'd be really surprised, about what you learn and then the people you meet. And that's what I kind of think is a big key to how I got to at least where I am, just by kind of taking some opportunities I kind of would have maybe thought, oh no, I wouldn't be interested, or oh, I wouldn't want to go by myself. But then when I pushed myself, it was really big opportunities to me.
Yeah, I'd agree. So there's two things. One is to obviously look for advertised opportunities with PhDs or master's programs, but I would also say that there's a lot of work that needs to be done to fill research gaps for bats that actually could be done by volunteer projects, or I know a lot of brilliant bat workers doing amazing stuff in their own time, and now that we have stuff like molecular techniques available, it becomes a lot more possible to do things. So one of the things we did with the population review process was just to highlight some of the really big data gaps and things, ways in which people can get involved and help.
Perfect, thank you. Thank you so much. Thank you to Fiona Mathews and Megan Power again. Really interesting talks. And thank you for everyone who's joined us this afternoon. Next week, we're actually going to go a bit different, and we're going to look at the conservation of wall paintings in churches, which promises to be a really interesting session. We've also got a few questions coming in about bats in churches and the challenges and solutions. And we're hoping to address some of these questions in a session on the 3rd of June, again at 1 p.m. And that's on a Wednesday as well. After this session, you'll have the opportunity to give your feedback. And if you can spend 1 or 2 minutes just filling that in, we'd really appreciate it. And it will help us make these events really good and exciting in the future. This was really, really good. Really enjoyed today. I hope you did too. If you want to see this again, we have recorded it, and we will make it available from our website, which is batsinchurches.org. Thank you again, and I'll see you next Wednesday. Thank you.